All right, lecture 10 is going to go over the urinary system. This is chapter 25, and the sections that are going to be covered are 4, 5, and 6 from this chapter. The urinary system obviously contains several organs. The main organ is the kidney, and the major functions of the kidney are to regulate the water volume and the solute concentration in the water, to regulate the ion concentrations in the extracellular fluid, ensuring the long-term acid-base balance, excreting metabolic waste like urea, toxins, drugs, producing erythropoietin. So this is a hormone that regulates blood pressure, and renin, which is involved in the production of red blood cells, then activating vitamin D. So vitamin D is produced in the skin, or it can be obviously taken uh, in food. But when it's in the body, it has to be converted into an active form. So the kidneys are the ones that convert the vitamin D into the active form. The other parts of the urinary system are the ureters. So the ureters are where, um, well, once the filtrate is filtered in the kidneys, it'll exit parts of the kidneys as urine. And then the urine collects and then is transported from the kidneys into the urinary bladder through these long tubes that are called the ureters. The urinary bladder is where the urine is held. It's stored, I should say, and then it'll exit the body through the urethra. So if we look at the inside of the kidneys, there's different structures here that are involved in the formation of urine. Most of the filtration is going to occur up here in the renal cortex, and a lot of the absorption occurs in the renal medulla, which is this inner layer. So when the filtrate is filtered, filtrate is just it's derived from blood plasma. When that filtrate is filtered and then it makes its way to the inner regions of the kidney, at that point it turns into urine, and then it'll collect through these uh, major major and minor calyces, and then into this renal pelvis, where it then goes into the ureter. But the structures that actually do the filtration are called nephrons. So these are structural and functional units that form the urine. There's about a million per kidney, so there's a lot of these tiny units, and they have two main components. There's the renal corpuscle and the renal tubule. The renal corpuscle has two parts on its own. So it's made up of the glomerulus, which is this essentially it's like a convoluted structure of capillaries. Looks like a ball of yarn of capillaries. And this is where the filtrate is formed. So the blood plasma that's being transported by the arteries arrives here at the glomerulus, and that's where the plasma is turned into filtrate. And then there's the glomerular capsule. It's also known as Bowman's capsule. It's the structure that surrounds the glomerulus. Then there are the tubular components to the nephron. There's the proximal convoluted tubule. So this is the tubular portion that's closest to the glomerulus. That's why it's called proximal. There's the nephron loop, also known as the loop of Henle. And then there's the distal convoluted tubule. So it's the distal convoluted portion um, that's farther away from the glomerulus. And then that distal convoluted tubule uh, merges into the collecting duct. Because again, there's millions of these. There's not just one. And these are mainly located in the renal cortex. Although some of the components do um, go into the inner layer, into the renal medulla. So there are three processes that are involved in the urine formation. There's glomerular filtration, which produces the cell and protein-free filtrate. There's the tubular reabsorption, which is what selectively returns most of the substances from the filtrate to the blood and the renal tubules, and also the collecting ducts. And then there's the tubular secretion, which is selectively moves substances from the blood to the filtrate in the renal tubules and collecting ducts. Okay. So the blood is obviously traveling through the arteries. So in this case, it's the cortical radiate artery. It reaches the glomerulus here. So this is the network of capillaries that are fenestrated. So it allows for substances to, or it allows for the filtrate to exit the blood vessels, but it keeps the red blood cells in here. So the red blood cells do not exit the glomerulus. They continue to the efferent arteriole. Okay, so this one's called afferent arteriole because this is where the blood arrives at the glomerulus. And then there's an efferent arteriole, which is where the blood exits the glomerulus and returns to the circulatory system. But the filtrate, the stuff that exits the glomerulus, is what eventually turns into urine. So once the filtrate exits the glomerulus or this capsule, then it's considered urine. As part of the filtration, there's several components that participate in that process. So there's the filtration membrane. It's a porous membrane between the blood and the interior glomerular capsule. This allows for water and small solutes to pass, but it doesn't allow for the red blood cells to pass. And it contains three layers. There's the capillary endothelium. That's a fenestrated endothelium that allows for blood components to go through, but again, not the actual red blood cells. Then there's the basement membrane, which also acts as a filter. And then finally, the last line of defense is the foot processes of the podocyte of the glomerular capsule. So these also prevent substances that, like large macromolecules that might have slipped through, it prevents them from going any further. There's also different pressures that affect filtration. So there's outward pressures. These are the forces that promote the filtrate formation. So this is essentially, for example, the hydrostatic pressure. That's the pressure inside of the, the glomerular capillaries. So that's pushing the essentially it's pushing the water and the solutes out of the blood. And then there's the inward pressures, like the hydrostatic pressure in the capsular space. So the glomerular capsule is this whole structure. The capsular space is the space within that capsule. And then there's the osmotic pressure in the capillaries, which is created by the proteins in the blood, which draw, or you could say suck water into the capillaries. So because pressure does move down the pressure gradient, or substances move down the pressure gradient, that also prevents filtrate formation here in the inward pressures, because the hydrostatic pressure in the capsular space, which is out here, is going to be higher than it is inside of the glomerulus. And so it prevents the filtrate formation because it's moving in this direction, back inward. In the outward pressure, for the hydrostatic pressure of the glomerular space, the filtrate inside is at a higher, it's under higher pressure because the capillaries are, they have a smaller space. So when you, or I should say, a smaller volume. So when it has a smaller volume, as it's under higher pressure. So here the filtrate is under higher pressure inside of the capillary, so the filtrate would move out down the pressure gradient into the capsular space, which is under less pressure. And then there are regulations to the glomerular filtration. So the glomerular filtration is important because it allows for the kidneys to make filtrate and maintain extracellular homeostasis. And there's two types of controls. There's intrinsic controls, which regulate at the kidney level, and this type of control maintains the glomerular filtration rate in the kidney. The glomerular filtration rate and also affects the systemic blood pressure because an increase in the glomerular filtration rate increases the urine output. And if you're increasing the urine output, that's going to lower the blood pressure. And it also works the other way. Decreasing the glomerular filtration rate causes a decrease in urine output, and it's going to increase the blood pressure. And the intrinsic controls, those are maintained by the nervous system and also the endocrine system. So there are hormones that act as intrinsic controls. So if we look at one of the intrinsic mechanisms using this chart, this flow chart. So here the situation is that there's a decrease in systemic blood pressure. So these are ways that the body responds to that. And they're grouped into two categories, intrinsic and extrinsic. But if we look at one of these, so we have low blood pressure, low systemic blood pressure, I should say. So that's going to lead to a low blood pressure in the afferent arterioles, because remember, afferent arterioles are the ones that bring the blood to the glomerulus. So that's where how the blood arrives. So there's going to be low blood pressure in the afferent arterioles. That's going to mean low glomerular filtration rate. That's going to cause a stretch of smooth muscle in the walls of the afferent arterioles. And then you're going to have vasodilation of the afferent arterioles. Vasodilation meaning that the diameter of the blood vessels increases. And so when that happens, you have an increase in the glomerular filtration rate, which is going to increase the blood pressure. For the extrinsic mechanisms, one way that could happen is these cells that are called granular cells at the juxtaglomerular complex of the kidney. And so those cells are going to release renin, which is a hormone that I already mentioned. And one of the functions of renin uh, is also involved in blood pressure. So renin is going to form into angiotensin 2. Angiotensin 2 is going to lead to, it can go either way, but we can choose this side. With the angiotensin 2, that's going to lead to vasoconstriction of the systemic arterioles. And that's vasoconstriction is going to lead to an increase in the peripheral resistance. So that's going to increase blood pressure. So vasoconstriction is, just reducing the diameter of the blood vessel. Okay. And if you have a smaller window through which the fluid can run through, that increases the pressure. Okay, so increase in systemic blood pressure. So you don't have to know all of these, just know the ones that I went over. Okay. Step 2 in the formation of urine is tubular reabsorption. Tubular reabsorption reclaims most of the tubular contents and returns them to the blood. Okay. So here, this is part of the tubular structure of the nephron. Here's the glomerulus. Tubular reabsorption would be where those substances are returning to the circulatory system. Because this is the afferent arteriole that leads into the glomerulus. This is the efferent arteriole where the blood exits the glomerulus and returns back to the circulatory system. So for tubular reabsorption, that's where substances are reabsorbed back into the circulatory system. Almost all of the organic nutrients are going to be reabsorbed. And also water and ion reabsorption is hormonally regulated and adjusted. So it includes active and passive tubular reabsorption. Tubular reabsorption is going to occur by transcellular and paracellular routes. Transcellular is when the water and the solutes move, across the cells. Paracellular is when they move between the cells. So if we start here, there's going to be either water or solutes moving across the apical surface of the membrane. Apical meaning like the top. Then it diffuses through the cytosol, the inside of the cell here, or the fluid portion of the inside of the cell. And then it goes across the basolateral membrane. It enters into the interstitial fluid. And then from there, it makes its way into the capillary. So sodium is the most abundant cation in the infiltrate. And the main way that it's transported is by primary active transport. Secondary active transport is used by organic nutrients that are reabsorbed. So that would be things like glucose, amino acids, and vitamins. And they're co-transported with sodium. So here we look at the active transport of sodium that occurs here with the sodium and potassium ATPase. So that's where it's pumping 3 sodium out of the cell and 2 potassium into the cell. When it does that, it creates a gradient for sodium to enter from the filtrate and the tubular into the cell. Because as you're moving sodium out of this cell, that means that there's less sodium inside of the cell. There's more sodium outside of the cell. So sodium moves down the concentration gradient and it's going to move into the cell because of the gradient created by the sodium and potassium ATPase. And then there's going to be along with sodium, there's going to be reabsorption of organic nutrients that are going to be co-transported uh, through the apical membrane of the cell. There's also reabsorption of water by osmosis through the aquaporins. So aquaporins are little tiny pores, little proteins that create pores that allow for water to cross. And so water reabsorption increases the concentration of the solutes that are left behind, and these solutes can then be reabsorbed as they move down their concentration gradients. So that would be like lipid-soluble substances or various ions and urea. Passive tubular reabsorption of water, that's when the movement of sodium and other solutes creates an osmotic gradient for water. Because remember, water always follows the higher solute concentration. The water is going to be reabsorbed by osmosis, and it's going to be aided by those tiny pores that are called aquaporins. Passive tubular reabsorption of solutes, so that would be like the fat soluble substances, ions, and urea, like I mentioned, where they flow or where they follow water into the peritubular capillaries when they're moving down their concentration gradients. That's one of the reasons why lipid-soluble drugs and also some other toxins are reabsorbed even though, it's not desirable. So that's talking about here. When water moves through these aquaporins, it leaves the, the substances at a higher concentration here. So then that means that they move down their concentration gradients and follow the water back into the capillaries. So in the proximal convoluted tubule, that's the site for most of the reabsorption. 65% of the filtrate is reabsorbed here. So that's mainly water, sodium, bicarbonate, and other ions, and also organic substances like glucose, amino acid. In the descending limb, this is where water could be reabsorbed. Solutes can't. Then there's the ascending limb of the nephron loop where water cannot leave because of the absence of aquaporins. So it just can't It can't leave the tubule, but solutes can. And then there's the well, there's yeah, we'll get to the distal convoluted tubule. The distal convoluted tubule and the collecting duct. This is where reabsorption is hormonally regulated. So, for example, at the distal convoluted tubule, salt is regulated by the hormone aldosterone, and calcium is regulated by the parathyroid hormone. There's also the ADH, which is the antidiuretic hormone that it affects the how much water is being reabsorbed at the collecting duct. Because when ADH is released into the blood, it causes the cells of the collecting ducts to insert the aquaporins. And so this increases the water absorption. So more water is reabsorbed back into the body and less water is produced as urine. Or there's less water that leaves the body as urine. Okay. So things that could trigger the ADH could be like exercising heavily. Like if you're doing a lot of exercises and you're sweating a lot, that could trigger your hypothalamus. What will signal the hypothalamus will tell the posterior pituitary gland to release the antidiuretic hormone. And when that reaches the kidneys, again, that's going to allow for the insertion of aquaporins. So that means that with more aquaporins, more water is going to be able to be reabsorbed back into the body, and less water is going to leave with urine. So that's why when you're dehydrated, you produce less urine because when you're dehydrated, there's a release of antidiuretic hormone, which causes more water to be absorbed. If you're fully hydrated, then obviously there's not a lot of ADH, so more water will leave in the urine. There's also things that affect the antidiuretic hormone, like alcohol and caffeine. Those two inhibit antidiuretic hormone. So when antidiuretic hormone is inhibited, that means that there's less aquaporins that are here at the collecting duct. So that means that uh, more water leaves with the urine. So that's why when you either drink a lot of caffeine or alcohol, you tend to want to urinate more, or you produce more urine. Tubular secretion is going to be reabsorption, but in reverse. So here we're going to focus on the green squares, because that's where substances are moving from the blood vessels. Because something you should keep in mind is that there are blood vessels here running over the nephron. So this is where the substances are secreted by the blood vessels and then moved into the filtrate to be excreted by the tubular component of the nephron. So substances could be like potassium, hydrogen ions, ammonium, creatinine, uh, or bicarbonate. That's another example. Okay. And the main thing for tubular secretion is to dispose of the substances like drugs or metabolites that are bound to the plasma proteins, and also eliminating undesirable substances that were passively reabsorbed, like urea and uric acid. Okay, so that's going to be it for this chapter. So I already mentioned one of the long response questions, which was the cross bridge cycle from chapter 9. The other long response question is going to be the changes in the thoracic volume, or I should say the sequence of events during inspiration and expiration. So you do have to know how the thoracic volume changes, how that affects the pressure inside of the thoracic cavity, and what muscles are involved in either increasing the volume or decreasing the volume.